Hodgkin's lymphoma has demonstrated to be an exquisitely sensitive disease to immunotherapy. There are 2 approved checkpoint agents that have shown high clinical activity in this patient population. The tumors in Hodgkin's lymphoma do express high levels of PD-L1 and PD-1, both in the tumor and the microenvironment, suggesting a possible target for checkpoint inhibitors. In addition, very elegant work done by Margaret Shipp in her lab has shown amplification and multiple copies of the gene for PD-L1 and L2 on chromosome 9. This may be a potential source for overexpression and may be a way to target patients and tumors in the future for checkpoint inhibitor therapy.
We're looking at ways to improve the outcome of patients over standard therapy. So a further approach has been to utilize a PD-1 blocking antibody, nivolumab, in combination with standard chemotherapy. The results with PD-1 blockade in relapse patients have been very exciting. High response rates, again very durable. Now it's being tested in the front line. So the cohort D of the 205 study was looking at using nivolumab in combination with AVD chemotherapy. This was a small study, only 51 patients, more of a feasibility study, and it showed that you can safely combine nivolumab with AVD chemotherapy. It also had an interesting window to it where patients received nivolumab alone first for 2 months and then received the combination. And that gave us some insights into the efficacy of nivolumab as the frontline first treatment, and it showed efficacy even as a single agent. I think it's going to take a randomized trial for us to really establish it as a frontline treatment. At this point, it's really still experimental and still in its testing phase, and so I don't think it actually impacts practice quite at this point. I think it's something to be watching for the future.
CheckMate 205 was a very important study because it looked at the use of nivolumab in Hodgkin's lymphoma in a variety of clinical settings. Most importantly, in the first 3 cohorts of the study, it looked at nivolumab in the relapse setting, either after brentuximab vedotin or after autologous stem cell transplant in a variety of ways. And in those cohorts, a high response rate and frankly a very durable response rate was seen. In the last cohort, known as cohort D, nivolumab was evaluated in the frontline setting. And this was the first ever instance where checkpoint inhibitor was evaluated in a curative setting with chemotherapy. And we learned a number of things. Importantly, I think we learned that nivolumab could be used in the frontline setting and is safe and tolerable. That is likely the primary take-home point from this. Secondly, however, we evaluated and have a better understanding of the single-agent activity of nivolumab in this setting due to the lead-in period. And there was indeed a response rate and a benefit seen in those patients who received nivolumab even prior to receiving the combination of nivolumab and AVD. And I think lastly, we have some appreciation now for the response rate of this regimen and curative potential, which we will learn as time goes on.